So I am the next speaker. Okay. And I will focus on splicing factor mutant MDS. The overall incidence of MDS is about 5 per 100,000 persons per year, but increases to more than 40 per 100,000 persons per year over the age of 70 years. So about 30,000 new cases are expected in Europe each year. And I must say that there is an ongoing project, MDS Right, focused on MDS, coordinated by Theo De Witte, who is in the audience. The last classification of MDS is shown in this slide. So we have MDS with unilinear dysplasia, with ring sideroblast, multilinear dysplasia, excess of blast, and MDS with isolated deletion 5q. And I will now focus on MDS with ring sideroblast. This is a bone marrow aspirate after pulse staining showing several ring sideroblasts. And these are immature red cells with pulse-positive granules surrounding the nucleus, and we know that these granules are iron-loaded mitochondria. The ring sideroblast must be distinguished from ferritin sideroblasts found in the normal bone marrow, in which the pulse-positive granules are just siderosomes, so lysosomes with aggregates of ferritin and hemosiderin. Most of ring sideroblasts die of excessive apoptosis in the bone marrow, so ineffective erythropoiesis is the mechanism of disease, and only few of these cells mature to red cells, circulating red cells, in this case called siderocytes, because they have the iron-loaded mitochondria for a few days. In order to define the genetic basis of this condition, we did whole exome sequencing in a European collaborative study, and found that of the 8 patients studied, 6 carried a somatic mutation in a gene called SF3B1, which encodes a splicing factor. And very strikingly, 4 of these 6 patients had exactly the same somatic mutation. Additionally, in 4 of the 8 patients, the somatic mutation in SF3B1 was the only mutation detected by whole exome sequencing, establishing a causal relationship between the mutation and ring sideroblast. More recently, Luca Malcovati has studied a population of patients with refractory anemia with ring sideroblast, and has shown that more than 90% of these patients carry a somatic mutation in SF3B1. SF3B1 is the only mutant gene in about half of these patients, and in most cases, co-mutant genes are TET2 and DNA83A. But these somatic mutations are found in the elderly, so they essentially reflect age-related clonal hematopoiesis. we have seen so far the genetic basis of MDS with ring sideroblast, but what about the remaining conditions? The genetic basis of all myelodysplastic syndromes is very complex, and I am here comparing two studies, one by Ellie Pape Emanuel published in Blood, and the second one by Torsten Affelak published in Leukemia. So altogether, here we have more than 1,500 patients, and in both studies, only 6 genes are mutated in more than 10% of MDS patients, and these genes are SF3B1, TET2, SRSF2, ASL1, DNMT3A, and RUNX1. So altogether, most patients have somatic mutations of RNA splicing machinery or DNA methylation. Of the 20 most frequently mutated genes in both studies, 17 are the same, so indicating that this genetic basis is really reliable. And finally, about 50% of MDS patients have somatic mutations in a gene of the spliceosome, indicating that splicing factor mutations are quite common. In a previous study, Seishi Ogawa showed that there are different genes of the spliceosome mutated in MDS, SF3B1, SRSF2, U2F1, ZRSR2, and with a few exceptions, these mutations are mutually exclusive. Let's just consider two of these genes, SF3B1 and SRSF2. So they both operate in 3' splicing, but the phenotypes of the disorder associated with these mutations are quite different. SF3B1 is associated, as we saw previously, with ring sideroblast, while SRSF2 is associated with multilineage dysplasia. So different splicing factors result in different phenotypes, but more importantly, they have very different clinical effects. Here, Luca Malcovati has done a study comparing the clinical outcome of SF3B1 mutant MDS with the clinical outcome of SRSF2 mutant MDS. As you can see, SF3B1 mutant MDS is a very indolent disease, while SRSF2 mutant MDS is a very aggressive disease. So, how can a somatic mutation in a splicing factor result in MDS first, and how can different splicing factors result in different MDS? Let's start with refractory anemia with ring sideroblast. We previously found that refractory anemia with ring sideroblast have a very distinct gene expression profile with overexpression of the gene called ALAS2 and decreased expression of a gene called ABCB7. These genes are important in mitochondrial iron metabolism. First, because SRSF2 is the first enzyme, ALAS2 is the first enzyme of the heme synthesis, while ABCB7 is the gene responsible for the exportation of iron from the mitochondrion to the cytoplasm for iron-sulfur cluster biogenesis. In order to understand what happens in SF3B1 mutant MDS, in collaboration with Seisho Gawa, we did RNA-seq studies on RNA extracted from CD34 positive cells and RNA from erythroid cells. So here we have ABCB7, the gene whose product is responsible for exportation of iron from the mitochondrion. You can see that there are two types of transcripts, one normal or canonical and the other one abnormal in cells from these patients. And the abnormal transcript is more clearly visible after the cells have been treated with cycloheximide. So cycloheximide prevents RNA decay, and this indicates that in these cells there is a spontaneous RNA decay of RNA from ABCB7 gene. And when we assessed ABCB7 in the cells of these patients, we found that indeed cells from patients with SF3B1 mutant MDS have about half the normal value, and the difference is very consistent. In order to understand what happens, first you should consider that SF3B1 binds RNA at the intron-exon junction, and then the interaction of the spliceosome leads to the excision of the intron and the splicing of the two exons. In a patient with SF3B1 mutant MDS, the mutation is always heterozygous. We have two types of splicing factors of SF3B1, normal splicing factor and in red mutant splicing factor. So of all splicing events, about half will be operated by normal splicing factors and about 50% by abnormal splicing factors. Silvia Bonamici, who is working in Boston for H3 Biomedicine, has shown in a cellular model that clearly the normal SF3B1 splices normally and leads to canonical transcripts and protein. But the abnormal splicing factor binds RNA upstream of the branch point, and this results in two types of events. First, frame shift, and second, to aberrant protein production or nonsense mediated RNA decay. So what happens in a MDS with mutant SF3B1 is that there is only one somatic mutation at DNA level, but several mutations, functional mutations at RNA level. And most of these mutations are frame shift events that lead to truncated proteins or to nonsense-mediated RNA decay, so to haploinsufficiency. The combination of these events leads to gain of function function at stem cell level, so proliferation of hematopoietic stem cells. For instance, one of the genes whose haploinsufficiency is important for this proliferation is NF1. And the combination of events leads to loss of function at hematopoietic precursor level. For instance, haploinsufficiency of ABCB7 is responsible for accumulation of iron in the mitochondria of the erythroblast and for excessive apoptosis and ineffective hematopoiesis. Are these observations relevant to treatment of MDS? We hope so, because preliminary studies have shown that while haploinsufficiency is responsible for proliferation of hematopoietic stem cells, it also implies that by using splicing factor inhibitors, these mutant cells with a somatic mutation in SF3B1 and the haploinsufficiency of several genes are more sensitive to the inhibitors. So by using these inhibitors, it is possible to selectively kill the SF3B1 mutant cells. And a protocol for a clinical trial based on the use of a splicing factor inhibitor has already been submitted to FDA for a clinical trial. I will finish just thanking all my collaborators in Pavia and the many collaborators we have worldwide. Thank you very much.